Okay, welcome everybody to our next episode of the Curious Circuit podcast. This week's episode is entitled The Story of Chocolate, and I'm joined here in studio by Dr James Richardson. James is a member of faculty in the School of Biological, Earth and Environmental Sciences in UCC. Welcome, James.
Hello, Barbara.
So James, I suppose just to begin, tell us a little bit about yourself and how you became interested in the source plant of what we know as chocolate today. Thank you.
Okay, so grew up in rural Oxfordshire, surrounded by nature, but ignored it for the first 18 years of my life. Went to university. I went to University of Leicester. And despite my ignorance of nature, I was actually interested in biology, so I did a degree in biological sciences. Although initially I thought that I might be interested in microbiology. I soon found that I wasn't. I wasn't interested in looking at petri dishes, but I got more and more interested in the plant-related subjects, so plant physiology, plant systematics. And so I ended up doing a fourth-year project, well, a final-year project, in taxonomy, and that involved basically looking at herbarium specimens, which we ordered from the Natural History Museum in London, and I looked at the morphology of those specimens in quite a lot of detail, so maybe about 150 specimens, to try to determine how many species there were in this particular group of plants that I was looking at. And what I also did was looked at the chemistry. So looked at the secondary chemistry to compare secondary chemistry and use that also as a way of defining or delimiting species. So different species would have different secondary chemistry profiles, so different qualitatively different secondary chemicals.
What plants were you looking at?
So this was a genus, bizarrely enough, it's from Chile in South America. Mm-hmm. It was called Francoa. And there was a question as to whether there were only 4 species or 2 species in the genus. So that was basically what I was trying to use all this morphological and chemical data to determine. And so I did that project. And then I was back home for a year trying to figure out what to do. But I was kind of interested in the taxonomy and more into sort of evolutionary biology. And I applied for a scholarship, which I'd noticed advertised in, I think, New Scientist, to do a master's in pure and applied plant and fungal taxonomy at the University of Reading. And so I went for an interview, and the guy that interviewed me was the world expert on chemotaxonomy. So it was just fairly fortunate for me, because I was obviously very interested in the subject that he was interested in. So anyway, so I got the scholarship. So I did that plant taxonomy master, plant and fungal taxonomy masters. And while I was doing that, I started using DNA as well. So I did another project, again looking at chemotaxonomy, but also starting to look at DNA sequence data using fairly sort of primitive technology for the time. And then I was invited to go and contribute to a book on European garden plants at the Royal Botanic Garden in Edinburgh. So immediately after my master's, I moved up to Edinburgh. I was working on that project for 3 years, trying to figure out what to do next, but sort of with a pretty good idea of what I wanted to do, which was to use molecular data, so DNA sequence data, to determine relationships amongst different groups of plants. And I got offered the chance to do a PhD at Kew Gardens. And when I started that PhD, it was at a time where the technology had really taken several kind of big steps forward in terms of DNA sequencing. So I was able to utilize this new technology to produce what for the time was a large amount of DNA sequence data. And so so my project was on a group of plants which were from South Africa. And I spent the first year working in the laboratory, trying to generate DNA sequence data from specimens from living individuals, but also using specimens from the herbarium. So I was one of the first people to extract DNA from herbarium specimens and use that DNA.
I think that's really interesting, James, to think that these specimens that are generally quite old in herbaria, that one can still get, DNA of sufficient quality for analysis. How old were the specimens you were working on? Do you know?
So I think the oldest one I worked on was about 100 years old.
Yeah, that's really interesting, isn't it?
So if the specimens are collected and dried quickly, then the DNA stays pretty much intact. So it depends on the method of collection.
Yeah.
So if they're collected efficiently and dried efficiently, then the DNA stays intact, which potentially means you've got access to millions of data points from millions of specimens. So the herbarium at Kew has 8 million specimens. The family that I was working on is what they call a cosmopolitan family, so it's found in all parts of the world. And so I was able to generate data from plants from Australia, South Africa, South America, all parts of the distribution of the plant, which I wouldn't have been able to do without access to that herbarium material. So those herbarium collections are absolutely essential. It's essential that we maintain them because we're always finding new uses for them as well.
I think that's a really important point. We might come back to that in terms of how those specimens can help us today in terms of climate and understanding spread of disease. Isn't that right? Yeah.
Yeah, absolutely. Yeah, it's absolutely vital that they're maintained. We're talking about collections, as I say, large numbers of specimens that have been collected over the course of the last 200-300 years. So absolutely invaluable resources for plant research.
Yeah. Okay, so that was you did your PhD in that space. What happened next?
Yeah, so I was doing my PhD in Kew Gardens. I was able to utilize all this new technology. And the group of plants that I was working on was a genus which consisted of about 150 species, which were found around the southwestern tip of South Africa. And it's one of the most species-rich places on Earth. And one of the questions is, why is it so species-rich? So some of my research kind of tried to attempt to resolve that question. And so I was able to get material of these species through fieldwork. So I did I think 3 field trips to South Africa. And another interesting thing about this particular genus is that there are species which are found on islands around southern Africa. So islands in the Atlantic Ocean, in St. Helena, in the Tristan da Cunha group, and in the Indian Ocean on New Amsterdam Island, Madagascar. And so that distribution was really interesting as well. And one of the main sort of the main focus of my research was just to try to determine how those species were related to the mainland ones. But I was actually able to extend that even further, because I was able to use the fact that we knew the ages of those islands in terms of they're volcanic islands. The majority of them are volcanic islands. They only emerged from the sea, at a particular point in time. And we could use that temporal information and apply that to the DNA-based phylogeny that they produced. So phylogeny, which is like a tree of life of all of the species that I was working on, using DNA sequence data. And we applied molecular clock techniques to determine when speciation events occurred. And what we found was that speciation was occurring over a period of time in that group when there was quite strong climatic changes. So basically South Africa was once pretty much ever wet climate. The climate was ever wet. And then as a result of a number of geological changes, tectonic changes, oceanic current patterns changed, and that affected the climate in southern Africa. So it basically developed a Mediterranean climate with very dry seasons and wet seasons. So the plants had to adapt to those dry conditions. And this actually led to a lot of extinction. So a lot of plants that were adapted to wet conditions didn't survive, so they went extinct. And the plants that remained, the lineages that remained, were able to speciate into the niches that were evacuated by these species that went extinct.
And could withstand drier conditions.
Yeah. Exactly. So again, like talking about, I'm extremely fortunate to be able to go and do some of my first real proper field work in one of the most species-rich places on Earth.
Beautiful.
In South Africa, which is absolutely astounding. Every place we went to, we'd be able to collect 7 or 8 species from my group of interest. And also, I had the possibility of going to one of these oceanic islands. So I went to the Tristan da Cunha group, which is a 5-hour trip from Cape Town on a ship, to collect this one species, the only species of tree that grew on the island, which was an absolutely astounding opportunity. And it was interesting not just from a biological and a research perspective, but also there are 300 people that live on the island. It's the most remote human population on Earth. 300 people, so the kind of the history of that group of people and just the sociology. So embedding yourself in that community and trying to understand how they function.
How long were you there?
Just 3 weeks.
3 weeks.
So at that stage, when I went, which is 1997, I think, there was only one trip per year, one boat trip per year. So I had to be very careful to make sure I got on that trip, because otherwise I'd have to delay the whole thing for a year. But very, very fortunate to be able to go to one of the most species-rich places on Earth, and then to a place which was very, very low in species richness, only about 100 species of plants on the island. And to be able to use those collections to determine the biogeographic history and the diversification history of this group of plants. And because I was using fairly new techniques at the time, new analytical procedures, we were fortunate enough to get that published in Nature.
That's an amazing feat during your PhD to get in Nature publications. That was really good for you.
Yeah, as I say, very fortunate, but and just fascinating, very interesting.
I'm thinking in my head here as you're talking about your trip to these lovely places in Tristan da Cunha. I'm thinking kind of similarities between you and Charles Darwin on the Beagle in terms of collecting samples.
Yeah, well, I think that the comparison should stop there, because I don't think there's any.
So, James, that's all wonderful, and you've painted a fabulous picture of your early years during your PhD of, building up your repertoire of techniques. You're at the coalface of DNA and sequencing and all of that, and your collections and these fabulously diverse parts of the world. It's just amazing, really. Let's kind of relate this to your current work in relation to the story of chocolate. So the importance of everything you've said, you're now using all those techniques and all of that, kind of those methods that you've kind of, fine-tuned during your PhD. And this led you to kind of move to a different species, isn't that right? A different genus?
Yeah, so basically, since I've finished my PhD, I spent maybe 10 or 15 years working on lots of other groups of plants, doing very similar things in different parts of the world, so in other parts of Africa and Latin America and Southeast Asia.
What was the connection between all of the different species? There was an underlying connection, I presume.
Yeah, so the basic message from all of those studies was that, as I said in the study I did for my PhD, there were groups of plants that weren't able to adapt to intense physiological changes, in this case, in particular to drought. And so we demonstrated that on lots of different groups of plants in different parts of the world, in different biomes, different ecosystems. And so we published a lot on that particular theme. But I arrived at a point where I was kind of frustrated at the fact that it was reaching the academic community but wasn't really going beyond that. So I wanted to do something that was of interest to more people. So at the time, when I kind of ended up reaching to that conclusion, I was based in Colombia, and I needed to make a choice about working on a group of plants, basically doing what I've been doing for the previous 20 years, on a group of plants that were interesting for people outside of the academic community. And so the obvious choice was to work on the tree that's the source of chocolate, which is Theobroma cacao, which is native to Amazonia and perhaps other parts of South America and Central America. And so basically I started working on Theobroma cacao and their relatives, doing exactly what I'd been doing previously.
Okay, so Theobroma cacao is the source of our chocolate.
That's the source of our chocolate. And so the first thing I did was to do what I'd been doing previously, which is try to determine the age at which that species and its relatives had evolved. And what we found was that Theobroma cacao as a species is actually quite ancient. It's about 10 million years old. And so we published this paper, and it got lots of publicity because of that fact, because people are interested in chocolate, and it was kind of an interesting kind of age. And so I got more publicity the week after publishing that article than I'd got in the previous 15 or 20 years. And so we want to kind of use it as a vehicle to try to explain why the research we're doing is important, and why looking at the history of these groups of plants is important to determine how they responded to climate change or geological change in the past, so we can maybe understand a little bit more about how they might respond in the future. So that's kind of the primary motivation of a lot of the research. But
so sorry to cut across you. Sometimes I think, and I know this from my own research and from various outreach activities, and I say we as the general public, we can take our plants for granted sometimes. Isn't that right? And we have this kind of, what we call plant blindness. And, we know, at least most of us know, the importance of plants in our daily lives. Think about what you eat, what you drink, what you wear, what you live in, the medicines you use. But sometimes we seem to have this blind spot as a species, as a human species, in relation to plants, the importance of them, what they need in terms of protection. You talked about adaptation, and nothing more important at the moment than one of our biggest challenges in terms of climate change, and how we can preserve our plants in the face of this, massive change that's occurring. In terms of chocolate, you have an interesting statistic about where we in Ireland fit in terms of consumption of chocolate.
Yeah, in terms of per capita consumption, Ireland lies third behind, I can't remember which is top, but Germany and Switzerland.
Yeah.
So in Ireland, the per capita consumption, the annual consumption is, I think, about 8.4 kilos, which I think is equivalent to, I tried to figure out how many Yorkie bars it was, but it was a quite phenomenal figure. But basically, the idea there is that although it's a tropical product, it's something that's really important to the economy of Ireland. It contributes to the economy of Ireland and a lot of other countries in Europe and North America.
And I think we could all relate to chocolate. And if we don't protect our source, we won't have our product. Isn't that right?
Yeah, exactly. So so one of the things we're trying to do is not only look at Theobroma cacao itself, but to look at the relatives of Theobroma cacao.
Yeah. Because
some of the issues that cacao faces are, climate change for one, in particular drought, but also disease, fungal and viral diseases. Yeah. So just to give an example, cacao or chocolate prices rose, I think, threefold at the beginning of last year, in the first 2 or 3 months of last year, as a result of low production, in part, but I think primarily because of drought in West Africa, which is the major producing area. So what we're trying to do is look at the biology of these related species, try to determine whether any of them might have evidence of drought tolerance or disease resistance, and whether we can use that information to improve productivity in cacao to make them more resilient or tolerant to disease or drought.
Can you tease that out a little bit for me? Because you've talked about relatives of Theobroma cacao. So are you saying in terms of cross-hybridization, in terms of breeding, or are you talking about using these relatives in place of Theobroma cacao?
That's a good question. That's a long question to have to answer. So basically, in terms of interbreeding, it's very difficult to interbreed species. So traditional breeding approaches probably wouldn't work. So what we're talking about is making slight changes to the genome of Theobroma cacao based on our understanding of the genomes of related species to make them more tolerant to drought or disease resistant.
So I guess here you're talking about, some people recognise the term CRISPR-Cas, gene editing, those kind of strategies, is that right?
Yeah, but we're talking about very subtle modifications. So we're not talking about taking a gene from a fish and putting it into a plant.
Yeah, no. The newer techniques like gene editing,
Yeah, so it's a very complicated story because drought tolerance involves large numbers of genes, and disease resistance probably also. But we're starting to look at basically doing comparisons between the genomes of cacao, for which we have quite a lot of information, with related species that might be able to help.
Okay, so that's one thing. And then go back to my other part of that question about using related species in place of or in addition to cacao.
Yeah, so that's another area of research that we're interested in. So One of the things that's involved in the chocolate making process is one of the first processes is fermentation.
Mm-hmm.
So basically the cacao beans, where you get the chocolate from, are fermented in a pulp which
Mm-hmm.
exists in the fruit. So you take out the beans with the pulp still intact,
Mm-hmm.
and then that pulp ferments the beans and chemically alters the beans so that they're more palatable. The chocolate is more palatable to us. So that's the first step in a very complicated process, but it's a very important step. And that fermentation process actually differs depending on which part of the area of cultivation that you're in. So the bacteria and the fungi that are involved within the fermentation process will differ.
By geographic region.
Geographically, yeah. So and that will alter the flavor profiles. So that's where you get differences in flavor profiles, and the differences are like really quite stark. So it's almost like wine tasting.
That's really interesting. Has much work happened in terms of the microbiome associated with the fermentation process?
That's actually one of the things that we're interested in looking into. So we've been talking about the possibility of developing projects in that. So that's a big potential, I think.
Yeah.
So the pulp. So the pulp was first used by, the fruit of cacao was first used by indigenous people, in which they take a fruit pod into the forest with them. They'd cut it open. They'd eat the pulp and they'd spit out the beans. So the beans weren't actually, the process of developing, of making chocolate is actually very complicated, and it took a long time to figure it out. But the first use was just eating the pulp. So the pulp is potentially an additional product, but it's currently involved in the fermentation process. But if you can separate the pulp from the beans and somehow ferment those beans without the pulp, you've potentially got two products for you. Mm-hmm. Two products from Theobroma cacao, which is the species where you get chocolate from, but there are about 35 other species from which you could maybe do the same thing. Mm-hmm. So there's the potential to develop novel products in all of these other species, which is another thing that we're very interested in looking into, because I think it has tremendous potential. So a lot of these species are used locally to produce chocolate-like products.
Chocolate-like products. Have you tried the pulp yourself?
I haven't actually. No, amazingly. One species, related species, is already being used just for the pulp. So there's this thing called cupuaçu, which is an Amazonian species, and there's already an industry around that. So they don't really use the beans to make a chocolate-like product. They just use the pulp to produce fruit juices.
To produce fruit juices.
Yeah, or flavoring for ice cream. So,
but not chocolate flavoring.
Not chocolate. It doesn't have anything to do with any other flavor I've ever tasted, and I have had that, and that is totally unique flavor and very nice. So if you go to like a restaurant in a major city in Colombia or in Brazil, you'll be able to get cupuaçu. And it's like, I guess it's sort of a bit citrusy, a bit appley, but really it's a kind of a unique flavor.
Okay, that's really interesting. We can't get it in this part of the world. It's not imported.
I think you can get some like, you can get dried products, I think.
Yeah.
But I haven't located any within Ireland at least. But yeah, it's so the potential is there to develop a whole bunch of other products. So the uses of these wild relatives is the potential is enormous.
Yeah, and of course we're not going to deviate now too far from chocolate, but you mentioned there the use of wild relatives. In my own work, we are interested in looking at, say, disease-resistant genes in wild relatives and introgressing back into our domesticated crops, because as we will all know, that through the domestication process, many of our resistance genes have been bred out, either deliberately or accidentally, in terms of flavour and all of that. So, the use of those wild relatives is really important, isn't it? And it's really important to preserve those wild relatives. Yeah,
absolutely. So a friend of mine that works at Kew is doing pretty much what I'm doing with cacao, but with coffee.
Yeah.
And so there are a couple of species that they currently use to produce coffee. But they're also under threat from drought. Mm-hmm. And what he's done is he's rediscovered a species that they had been using previously, which is drought tolerant and which has a very similar flavor profile. Mm. So it's already drought tolerant, similar profile, so it's kind of ripe for redevelopment. And so, as you say, it's really important to conserve all these related species, because we don't know when they're going to be, well, I mean, I already think they're useful, but, for a lot of other crops, we don't know whether they're going to be useful for looking at all these issues as either as alternative crops or sources of information that allow us to improve existing crops.
Yeah, and of course with new technologies, we won't go into too much detail now, but like gene editing, we already see the rapid domestication of wild relatives through that technology as well. So that's all very interesting. So in terms then of the Theobroma cacao, what's next for you?
So at the moment, we're trying to look at one particular species, which is in a genus which is related to Theobroma. It's a genus called Guazuma, and it has a species, Guazuma ulmifolia, which is already found in dry forests in Latin America.
How closely related or not is this to Theobroma?
So it's in the same lineage. Yeah. It probably diverged from Theobroma quite a long time ago, but it is a close relative, or it's one of its closer relatives.
And where do we find that growing?
So that's in dry forests in Latin America. Mm-hmm. So it's already adapted to drought, and we're looking at the genome of that species, trying to sequence the transcriptome to try and understand what genes are actually expressed, especially in response to drought conditions, so that we can compare that with the genomes that have already been produced for cacao.
For cacao. So, okay, just for the uninitiated, shall we say, what does that mean in practical terms once you get this information?
So basically we could try to determine what genes are involved in the drought response in Guazuma, and try to look for related genes in cacao to determine how different they are, and to determine whether we can modify them a little bit to make them
drought tolerant.
Yeah.
Is Guazuma used commercially?
No, it isn't really.
No.
The fruits are completely different. They look completely different. They're actually eaten. They're a bit like granola. It's a bit like eating granola. So it's like a dry fruit, more of a dry fruit. It dries up quickly.
So eaten by locals, is it, in that part of the world?
Yeah,
yeah I know. Okay.
Yeah. So, and one related species is actually used, they grow plantations of it in Peru, and they use it in construction. So there's a huge number of potential uses of related species related to cacao.
Yeah.
And one of the interesting things, or one of the surprising things to me, was about the fact that despite the fact that this is a economically important group, I worked with a PhD student from Brazil who was doing a taxonomic monograph of all the species, so producing a description of each of the species, which involved the use of herbarium specimens.
In Theobroma?
In Theobroma.
Yeah.
And he actually, as part of his work, he discovered 3 new species of Theobroma.
Brilliant.
Which to me is absolutely extraordinary.
Yeah.
So these aren't species which were described based on collections that were made recently. They were described based on herbarium collections which might have been made over the last 20, 50, 60 years or so. So again, it demonstrates the importance of herbaria in allowing us to properly describe the full extent of the biodiversity of cacao wild relatives, and of any group of plants, but any group of crop wild relatives as well.
Yeah. So those 3 new species, that's amazing, isn't it, that were discovered in herbaria. They can be tracked back to in situ and where they're growing?
Yeah, so we know more or less where they grow.
Where they grow, yeah.
A lot of these species are going to be very restricted in their distributions, and probably quite threatened with
extinction.
Yeah, either through deforestation or climate change. So that's why we're doing our best to document them as well as possible.
Just in terms of Theobroma cacao and disease, I don't know, have you worked much in this space, but do you know in terms of threatened with diseases, is it a massive problem for Theobroma?
Yeah, it's a huge problem. So basically, depending on where you are, cacao plantations will encounter different diseases. So the diseases which are native to Latin America, and which are the biggest problems in Latin America, there are basically 2 species of fungi which are closely related to one another. And their common names are the witch's broom and the frosty pod rot. And witch's broom is a particular problem and pretty much wiped out a large part of the cacao industry in Brazil in the late 1980s and early 1990s. And it was actually considered such a problem sociologically that they actually made like a soap opera about it.
Wow.
To describe this issue of witch's broom. And so, there's a lot of research which has been done on trying to resolve that problem, but it's very, very difficult. It's a really tenacious disease.
Yeah. So still a real issue today. Yeah.
In Latin America. And then
the soap opera didn't
I don't
give any clues to it.
No, the soap opera didn't help much with it.
So I guess on a serious note, what is it, sprays, chemicals that are used to treat?
Well, so they use, I think they use copper hydroxide, actually. I'm not sure exactly how effective it is. The other way is just to prune off the diseased branches, but that's
catch it early and eliminate where possible.
But if you miss it, it's
real problems. Yeah.
And the diseases in West Africa will be different. So you don't have necessarily problems with those two species of fungi, but you have other
issues.
diseases.
Yeah, I mean we talk about the arms race, don't we?
Yeah.
In terms of disease, trying to stay one step ahead.
Yeah. So new diseases developing. So in Africa they have problems with Phytophthora, and they have problems with viral diseases as well. So
Phytophthora affects Theobroma?
Yeah.
Okay.
Yeah.
And our listeners may not recognise the name, some people at least. Anyway, Phytophthora, a species of Phytophthora, of course, wiped out the potato in Ireland in the 19th century. That's known as late blight, common name. Okay. James, that's brilliant. We look forward to seeing you at various festivals around the country, talking about the story of chocolate. You've done quite a bit of outreach, I know, in your time, and you're very passionate, I think, about bringing the story and the science behind chocolate to audiences. Isn't that right? Yeah,
no, I mean, the strategy of working on chocolate has definitely worked. And it allows me to interact with members of the public and try to describe the importance of some of the research we're doing, which I think is very important.
Really very important. And in fairness, who doesn't like chocolate? I challenge any listener.
Well, every time I give a public lecture, I ask if anybody does not like chocolate, and I haven't had one hand raised yet.
That's
so
it. Good broad audience there, James. Okay, so in typical style of another programme in the UK, Desert Island Discs, I want to finish by asking you to tell our listeners, if I were to restrict you to one of these lovely islands you've been to bring a piece of music, a book and a film with you, what would you tell our listeners?
Okay, I'd start with a film because I'd need something to make me laugh, and that would be With Nail and I.
With Nail and I, excellent. Starring Richard E. Grant. Super film, well done. Yeah,
it's an absolute classic and just hilarious every time. It makes me laugh. The book. So I would choose this book that I started reading about 10 years ago and got about 1/10 of the way through. It's called Mason and Dixon. It's by an author called Thomas Pynchon, and it's written in kind of an old English style. So it's a little bit difficult to read. So you have to read each page about 3 times, but it's very, very dense in terms of its content. So I only got 1/10 of the way through it. So I'd want to take that with me and finish it because I think it's a bit of a classic.
And a bit of
even
a
though I didn't manage to finish it. And then the music. So
wait 1 sec. Give us just a bit of background on the book. Should I go out and buy it? Tell us briefly about it.
So basically it's about Mason and Dixon, who are these 2 prospectors who basically mapped out large parts of northwestern North America.
For what purpose?
Well,
you don't know. You haven't read that much.
For exploitation.
Okay. Well,
and then
nice light read there, and a piece of music.
And so I'm only allowed 1, which is really.
Yes, 1 piece of music, James. You're travelling lightly. You may be taking Ryanair to your desert island.
Desert island
travel lightly.
Allows 8, but I've only got 1. So I'll choose this one. It's a bit of an obscure band called Television, who produced this album called Marquee Moon in the late 1970s, which is a classic guitar rock album. and the title track Marquee Moon, which is about 10 minutes long. So I'll choose something long.
Marquee Moon, okay.
And it's
noted. Must listen to that later on there.
fantastic.
I want to say a big thank you for joining me in studio today. And that is the story of chocolate. Thank you to everybody listening. Thanks, James.
Thanks, Barbara.